Okay, so I wanted to show you how to assemble your polypeptide, and I have a sample of DNA sequence. Yours will look a little bit different because yours will be on your function finder sheet and will be quite a bit longer. First, though, I've got my DNA sequence. You will have transcribed that, finding the complementary nucleotide that goes with the DNA nucleotide. And notice how I've got a U here instead of a T because uracil nucleotides replace thymines in RNA. So I've transcribed this particular sequence and produced my messenger RNA strand. And now I'm ready to translate it. So using the codon wheel, I will have translated this to which particular amino acid goes with this 3-letter sequence codon. And in this particular case, my polypeptide is 3 amino acids long, and I have valine, glutamine, and isoleucine. Okay, so now really it's about putting it together. I have all of the 3 papers that I'm going to need. And what's happening here is a chemical reaction. It's a fairly complicated process that we're not spending as much time on, but we want to be able to see how these particular monomers, these individual amino acids, are combined to produce a polymer, which in this case the polymer is a growing polypeptide. You can see that there are some dotted lines here, and what's going to happen is that you are going to be connecting these pieces. And notice how I'm only cutting the portion where I'm going to be connecting the amino acids. So my sequence says that valine is going to be first, so here's my valine paper. Here, the next one's going to be my glutamine paper, and I'm going to line up these lines here so that they appear to be bonded together. And in this case, what's called a carboxyl group, this double bonded oxygen with this hydroxyl group, this oxygen and hydrogen, are going to be linking together with where there was an amino group, which is this nitrogen with the 2 hydrogens attached. All right, so now I'm ready to tape those 2 together. I'll do the best I can to line them up. All right, now I do have some other materials though left over. I've got the hydrogen that was from the glutamine, and I have the hydroxyl from the valine. Those 2 are going to be now bonded together, and I'm pretty sure you know the name of the molecule that is HOH, otherwise known as H2O. I'll line those up, and I'm going to tape them on, and I'm going to add a little plus sign out here in the front so that I can recognize that when I bond 2 amino acids together, I also produce 1 water molecule. All right, I'm ready to attach now my isoleucine, the next one in the sequence. In the process in the cell, translation of the messenger RNA happens out in the cytoplasm, and a ribosome is required for this process, and also a different form of RNA called tRNA. All of that we're not showing. So what you are essentially pretending to be is a ribosome that's holding all the pieces close together so that they can be linked. I'll line this up the best I can. Take my H and OH, put them together. Put another plus sign to represent that this is another product. So the product that I'm building is my polypeptide, in this case 3 amino acids, and notice how in that process I have produced a couple of water molecules. Super important. Notice how I didn't cut off the hydrogen over here on the left on my first amino acid that was placed. And I will only continue cutting and adding as my sequence enlarges. When I'm all done and I've placed my last amino acid, I'll leave this part intact. All right, if you have any questions, of course you're welcome to ask as we're going along, and you'll be producing this in class. Thanks. Bye.